Welcome to Brain Science, the podcast that explores how recent discoveries in neuroscience are helping unravel the mystery of how our brain makes us human. I'm your host, Dr. Ginger Campbell, and this is episode 197. Last month's episode was on the technical side, so this month we're doing an episode that is somewhat aimed at newbies. I will be talking with my friend Frank Amthor, who is the author of both Neuroscience for Dummies and Neurobiology for Dummies. He's also the co-author of an excellent introductory textbook called Essentials of Modern Neuroscience. This episode is also a great review for longtime listeners. It's actually a free Encore episode from my premium back catalog. Before we jump into this month's interview, I want to remind you that you can get complete show notes and episode transcripts at brainsciencepodcast.com, and you can send me email at brainsciencepodcast@gmail.com. You can also get show notes automatically each month when you sign up for the free Brain Science newsletter. You can sign up at brainsciencepodcast.com or just text Brain Science, all one word, to 55444. That's Brain Science, all one word, to 55444. When you sign up, you will also get a free gift entitled 5 Things You Need to Know About Your Brain. Because Brain Science is independently produced, it relies on the support of listeners like you. You can learn more at brainsciencepodcast.com/donations. After the interview, I will be back to review the key ideas and make a few brief announcements. My guest today is Dr. Frank Amthor, author of Neuroscience for Dummies and his new book, Neurobiology for Dummies. Frank, it's great to have you on the Brain Science podcast today.
Thank you for having me.
And I think you might be the first guest that I've ever had on the show that was somebody that I knew outside of the Brain Science podcast. Did we originally meet at Dragon Con?
Well, we've crossed a few times on the tennis court, but
I don't remember which one came first.
Yeah, I don't either.
So, Frank, why don't you tell my listeners a little bit about who you are?
Okay. Well, I have a little bit of an unusual background in neuroscience in that originally I was an electrical engineer at Cornell University where the perceptron brain model was developed. And I became very interested in that model and interested in the fact that the computer that exists between everybody's 2 ears is in many ways way more powerful than any computer that humans can build. So I drifted from electrical engineering to artificial intelligence to neuroscience to the visual system. My interest in the visual system is kind of its circuits and systems, as you might expect of an engineer. I'm interested in how neurons do computations, how circuits of neurons do computations, and how we could restore the loss of function, for example, with a prosthetic eye, and maybe how even we could use brain principles to make better computers.
Wow, I don't think I even realized that was your background. How did you end up in the Department of Psychology at UAB?
I came to UAB as a postdoc in the School of Optometry doing research on the retina, and I had a grant funded to try to identify the basic cells in the retina, which are the retinal ganglion cells that project to the brain. These are, after all, the basis of visual perception because there are many classes that extract different kinds of information from the world. And a position open in psychology, and I applied for it because my interest is pretty much at the systems level more than, say, the cellular and molecular kind of level. So I started teaching courses. You know, even to this day, I'm very interested in where these cells project in the brain and what sort of visual behaviors that they subserve and those kinds of issues, which are very relevant to psychology.
Okay, that makes sense. So as I was preparing for this interview and even reading Neurobiology for Dummies, and I went back and forth to your first book, Neuroscience for Dummies, for overlapping material, and I have to admit that I'm still a little confused about what the difference is between neuroscience and neurobiology.
Well, you're right. There's a lot of overlap. Most people think of neuroscience as being a little more systems-oriented and perhaps a little more disorder-oriented. whereas neurobiology pays a little bit more attention to genetics and intracellular processes like second messenger systems and things having to do with biochemistry generally. Although in the limit, when you look at the visual system and so forth, they're really pretty similar there. Neurobiologists look at disorders from the point of view of what's the genetic basis, what's the mutation that creates this neurological disorder, whereas neuroscientists might look more at disorders that are caused by environmental conditions that don't necessarily have a clear molecular sort of basis.
Okay, so how did you end up writing these books?
Interesting question. Well, I wrote a science fiction novel called The Phoenix War, and I pitched this at an editor that I happened to come across from another faculty member. And the editor said, well, I don't do fiction, but I do nonfiction. Would you be interested in writing a nonfiction book if one came along that was suitable? And I said, well, that sounds like an interesting idea. So the second one that she ran by me was Neuroscience for Dummies. So we went through the process of submitting a table of contents and a sample chapter and ended up getting the contract to write that. And then a couple of years later, we did the same thing for Neurobiology for Dummies. So I kind of backed into it without really having long-term intention of writing nonfiction, but it was kind of fun, and I'm glad I did it.
So did you have, when you got ready to write the second book, in mind that there were things you hadn't covered that you wanted to cover, or was there an editorial board or something that was telling you what kind of content they wanted?
It was more the former. I knew that I wanted to cover more molecular issues and genetics. I had made a few mistakes in the first book, which people kindly pointed out by email, and everybody who did that was actually very nice about it. I'm, after all, not an expert on everything. So when I did the second book, I included a lot more material that was intracellular and biochemical and tried to fix the mistakes I made the first time and just do a better job writing it and so forth.
That's great, because I haven't really in recent years done much at the neurobiological level on the Brain Science podcast. I think whether I was aware of it or not, I have been more at the neuroscience or systems level, the big picture level. So it's nice to have an opportunity to return to some of the basic biology. And I thought that I would start out by asking you about neurons, since that's kind of the root of the difference of the biology of the nervous system is that's made up of neurons. So I thought I would start by asking you a basic but not that easy question of what is it that makes neurons different from other cells in the body?
Yeah, that's really getting at the heart of the matter. So neurons are specialized both in terms of their structure and then the function that that structure allows. The two major structural differences from other cells are they have what are called dendritic trees, which are branches that come off the main cell body that typically receive thousands of synaptic inputs from other neurons. And then they have an axon, which can go centimeters, inches, even feet in some cases, and contacts other neurons by carrying electrical impulses called action potentials down the axon to cause the release of neurotransmitters at the next neuron. So a human brain has about 100 billion of these neurons, maybe a little less, according to the latest figure, all communicating, firing action potentials, releasing neurotransmitter, processing sensory input, generating motor output, allowing us to execute all the behaviors that we execute and the thoughts that we have and so forth. None of this would be possible without these signaling capabilities that are inherent in individual neurons.
I want to talk some more about the signaling in a minute, but I want to focus on the action potential for a minute and ask you to expand a little bit upon why the action potentials are important. What makes them different from, say, other signals that might go on in the body?
So neurons in the nervous system generally probably evolved from secretory cells. Secretory cells, such as might be found in the hypothalamus and other places, will secrete hormones when they're exposed to chemicals in the environment or excess CO2 or something else going on in the body. These hormones diffuse away and they may bind receptors at some distance from the cells. what neurons invented by having an axon, they essentially could do their secretory process on a few very specific cells a long way away, skipping everything in between. This is essential, for example, in the control of muscles from the spinal cord, where you have neurons that go from the spinal cord to synapse on the muscles and cause individual muscles to contract. And neurons in the sensory system, which have specific receptor types that are sensitive to light or sound or pressure, that can send their signals to particular parts of the brain. And then very complicated computations can be done, like are done on a computer, to generate very complicated behaviors. There's just a limit to what you could do with a bag of cells all secreting their hormones in the general mix compared to what a system of nerve cells can do.
So targeting and distance.
Right, and specificity of that targeting.
Right. And I appreciated the fact that you emphasized in the book that nervous systems and brains in particular are evolved to support movement. It reminds me of a long time ago when I was talking to John Rady about the importance of exercise, and he told me the story of the sea squirts. You know the story of the sea squirts?
It eats its own brain, right?
Yeah, because it doesn't need to move anymore once it finds the place it's going to live. I want to take a moment to mention our longtime sponsor, Text Expander. What can you do with more hours every month? Repetitive typing, little mistakes, searching for answers. They're all taking away precious time. With Text Expander, you can take that time back so you can focus on what matters the most. You just type a few characters to trigger your snippet, and the content expands wherever you type. It's that easy. If you want to learn more about how to use Text Expander, you can go to textexpander.com/blog. I literally use Text Expander every day, and I really appreciate that Text Expander is available on Mac, Windows, Chrome, iPhone, and iPad. Brain Science listeners get 20% off their first year. Just visit textexpander.com/podcast to learn more. That's textexpander.com/podcast to learn more and get 20% off your first year. And don't forget to tell them that you learned about it on Brain Science. Okay, so let's talk a little bit more about the electrical and chemical signaling that goes on in the neurons. First, you mentioned hormones. So what's the difference between a hormone and a neurotransmitter?
Well, there's actually some overlap because some neurons release neuropeptides that are actually very similar to hormones. But the major difference is that there are important excitatory and inhibitory neurotransmitters that act very rapidly. The major excitatory neurotransmitters are glutamate and acetylcholine. And the major inhibitory neurotransmitters are GABA and glycine, and there are, of course, hundreds of them. But these open ion channels in the membrane of other neurons, or in the case of acetylcholine, also in muscles, that cause rapid electrical changes in those cells, which can cause communication and action potentials to be released in those neurons that go to other cells. So you can have very rapid and very specific computation. A given neuron may have several thousand excitatory and inhibitory inputs on it of 4, 5, or 6 different kinds. It's constantly and dynamically integrating the net balance of excitation and inhibition, which becomes reflected in the rate of action potentials traveling down the axon of that neuron, and therefore the rate of release of neurotransmitters onto the neurons that are postsynaptic to that neuron. And so each neuron is really a complicated little computer. And one of the things that caused me to move from electrical engineering to neuroscience was the realization that people in the early days of artificial intelligence were modeling neurons as switches, but they're far more complicated than switches. One neuron is more like a computer in of itself, whose activity is complicated way dependent on its past history, doing very complex computations on its input, and then shipping the output of that to other neurons. And then the brain has 80 to 100 billion of these neurons all doing this, so it's a very complicated system.
I know that some of the neuropeptides and other compounds in the body can sometimes be a hormone and they can sometimes be a neurotransmitter. What determines whether something's a neurotransmitter?
It's a neurotransmitter if it affects another neuron.
Interacts with a receptor?
Right. And what kind of cell it's a receptor on.
Okay.
So if it's neuron-to-neuron communication, whatever the substance is really a neurotransmitter then, even if someplace else in the body it's used as a hormone. I know a lot more about neurotransmitters than hormones, but as you said, there are substances that fulfill both roles depending on where they are.
So do you want to talk a little bit about the different types of receptors? Because that's really where the action's at.
There are a lot of neurotransmitters and a lot of receptors. So there are several hundred different receptors. And for each type of neurotransmitter, there are often 10 or 15 different receptor types. These receptor types can vary in how long the receptor stays open, what the adaptation time of the receptor is, the kinetics of the binding of the neurotransmitter. Some receptors called metabotropic receptors generate second messenger cascades within cells that may affect DNA expression in the cell ultimately and operate on homeostatic mechanisms. So neurons release neurotransmitters that may affect two different cells that are the receipt of the neurotransmitters in a completely different way. For example, there are dopamine D2 and D4 receptors that are believed to be important in schizophrenia. These are different than D1 and D3 receptors. There are various kinds of GABA receptors and acetylcholine receptors, and cells change the complement of receptor types that they express at different times depending on their past history of stimulation and what else is going on around them. So the nervous system is a dynamic system both functionally and, in fact, structurally because of the change in the complement of receptors that may be expressed. For example, if a neuron is being overexcited and receives too much excitatory input, it may begin expressing either more inhibitory receptors or inhibitory receptors that stay open longer, or if it's underexcited, it may do the opposite. And a lot of drugs that are used for things like depression and schizophrenia are drugs that affect neurotransmitter receptors or the action of different neurotransmitters on binding those receptors.
And since it depends on the receptor, you can't just say it's going to do one thing. That's one of the tricks of trying to design those drugs, I guess.
Right. You want to target particular receptor subtypes that are believed to underline the disease process in a particular part of the brain. Even if that neurotransmitter is used elsewhere, for example, in the eye, it may use a different receptor site, and you don't want to affect transmission in the eye. You want to affect only the receptor subtype that's involved in the disease process.
And our knowledge of that is still pretty primitive.
But it's improving all the time with having the genome sequenced and looking at slightly different receptor expressions of different people. An important area of pharmacology now is looking at different people's responses to different medications and being able to predict, possibly from variations in the different subtypes that some people express, the effectiveness or ineffectiveness of certain treatments. That's a very promising aspect of neuropharmacology now.
One of the things you talked about in the book that I found particularly interesting, and it was actually in the chapter about memory, but you talked about a particular type of receptor that's, I think, has been getting a lot of interest in some areas, and that's that Nmda receptor.
N-methyl D-aspartate, right.
Would you talk about that one briefly?
Sure.
Because that's kind of an unusual, or maybe not unusual, but interesting one anyway.
So people, even coming from the artificial intelligence community like I did, knew that there had to be some change in synaptic efficacy that underlied memory changes, the so-called memory engram. But 30 years ago, nobody really had any idea what that receptor was. There are probably a number of receptors that mediate memory processes, but clearly one of the most important ones is this Nmda receptor, which is found extensively in the hippocampus and also in neocortex and lots of other places in the brain. The Nmda receptor operates like an AND gate in a lot of ways, to use engineering terminology, which means that it's a glutamate receptor, and glutamate is the excitatory neurotransmitter that opens it. But when the cell is sitting there at rest at its normal resting potential, the binding glutamate alone isn't enough to open the Nmda receptor. Rather, the membrane has to be depolarized by some other glutamate input affecting some other receptor, like an Amthor receptor nearby. This causes the membrane to depolarize a little bit, releases a magnesium block in the Nmda receptor, and then when it binds glutamate, it will open. And so it's an AND gate in the sense that two conditions are required to open the Nmda receptor. One is the binding of glutamate, and two is the simultaneous depolarization of the membrane through another receptor. And then the other property of the Nmda receptor that's interesting is that in addition to fluxing sodium, like most excitatory neurotransmitter receptors, it fluxes a lot of calcium, which initiates second messenger cascades, which ultimately can affect the expression of DNA by the cell and the population of different receptor types. So the Nmda receptor can cause long-lasting changes in the cell, which can instantiate the synaptic plasticity necessary for memory. So if you think about when you need to remember something, it's usually a confluence of several things together. There's, something with a tail that's large, that has stripes. All of those things together indicate the presence of the tiger. And when all of those things activate the same cell, then you have a tiger detector, and that causes your memory to be triggered to not go near that water hole at that time of day or whatever it is. So the Nmda receptor is mediating a lot of the plasticity that supports memory.
So you started out in artificial intelligence and electrical engineering, so I would assume that means you have more than the average appreciation for what the difference is between brains and computers. Would you talk a little bit about that? You mention it often during the book, and it is an important concept that I keep trying to communicate.
It's something I think all people in neuroscience think about, but it's hard to get a handle on. One way of phrasing the question is, how important is the neural hardware of the brain, and particularly the details of the neural hardware? How essential is all of that for what the brain is able to do? The brain is able to do a lot of things, the human brain or even a frog brain, in terms of getting around and seeing things that computers aren't very good at. Of course, computers can do things that, we aren't very good at, like calculate pi to 100,000 places. So they have very different architectures. Computers work in serial. They tend to execute one process at a time, although they do this very fast. There's some parallelism in computers, but a human brain of 86 billion neurons is essentially operating entirely in parallel, and it's operating without specific programming. So people have asked the question, well, are action potentials essential? Could we simulate what the nervous system does by just virtually connecting the axon of one cell to the next cell without having the intervening action potentials? Or does the pattern of action potentials actually convey information? People had a lot of hope at one time that principles gleaned from the nervous system would be used to construct computers. So far, that really hasn't happened very much, although on the software side, there are artificial neural nets that are extensively used to do things like predict the stock market. And people may have heard recently about deep learning, where exposure to billions of instances of data, for example, Google has done this, in untrained hierarchical neuron-like systems that are simulated on computers, have the ability to extract patterns out of things like the internet. I think the first thing that the Google machine found was it could recognize pictures of cats. Like there are lots of on the internet without ever being told what a cat was or what it was supposed to look like. So that interplay between computer scientists and neuroscientists still goes on, and there's still fundamental questions. But the technology for making computer chips in silicon versus making neurons and brains is so different. There really hasn't been much closure there, except in the process of making neural prosthetics. And that's a field that I'm very interested in. There are hundreds of thousands of people who can hear now from prosthetic ear implants. And working on the visual system, it's my hope that we could do the same thing for vision, but we're really not very close to that yet. It's a much more difficult problem, requires hundreds or thousands of electrodes rather than 10 or 20. But, there are cases where we can make useful interfaces between computers and the brain. These are all done in the process of trying to alleviate some disability now, but in the future they may be done to actually enhance people. Imagine having an implant where you could go on the internet just by thinking about something that you wanted to inquire about, or you could have a question answered in your head by just thinking about it through some neural implant. Sounds like science fiction, but we're definitely developing a lot of the technology that would be necessary to do that in the near future.
It seems like one of the most obvious differences, although it wasn't obvious until we learned more about how memory really works, but our memory is very different from the way a computer memory is. Would you talk about that?
Sure. It's only beginning to be strongly understood now that human memory and animal memory, biological memory, is really essentially reconstructive. In a computer memory, a piece of data, some set of ones and zeros, is literally stored at some address. And when the memory wants to be recalled, the address is accessed, and that exact pattern of ones and zeros is moved to some buffer. But in the human brain, memory involves creating a context in the brain that's something like the context when the event actually originally happened, and essentially almost reliving the context. So one of the ways to understand this is to appreciate how the hippocampus, which we know is a memory structure, works with the neocortex, which is involved in processing sensory information. So the famous patient HM, for example, who had his hippocampus on both sides removed, had retained all his long-term memories prior to the operation, but was unable to form any new long-term memories. If someone came in and interacted with him and then walked out of the room and came back in 10 minutes later, HM would have no memory of ever having seen this person before. But his long-term memories were intact. And what we found out from that case and a lot of other cases is that memories are initially stored in the hippocampus, but then they're actually transferred to the parts of the neocortex that process the sensory information to begin with. So visual memory is stored in the visual neocortex, and auditory memory is stored in the auditory neocortex, and so forth. And so recalling things that have visual or auditory content or somatosensory or whatever involves activating the neocortex in a way that's similar to the way it was activated when the actual event first occurred. One of the interesting and important things about that is that we now know that one of the reasons that eyewitness testimony can be unreliable is that reliving a memory, particularly under powerful suggestion that's trying to point the memory in a certain direction, essentially rewrites the memory every time and can rewrite it into a state which is actually not right, but which you cannot distinguish from it not being right. Because you actually don't store the raw data, you store the ability to reconstruct something from the context. It took a long time for people to realize the reconstructive nature of memory, but I think it has really profound implications for not only neurobiology, but the court systems and a lot of other aspects of society.
So does that mean that if you had brain damage from a stroke that made you blind, that you would have trouble remembering what things look like?
People with damage to visual cortex have trouble with visual imagery. There's a famous textbook case, I think the lead author is Desiach, I'm probably not pronouncing that correctly, who had damage in his right visual parietal lobe. And the famous experiment was he was asked to imagine what he would see standing at one end of a famous Italian piazza, and he could describe all the things on the right side, but not the left side. And it's the left side of the visual world that projects to the right damaged side of the brain. When he imagined standing at the other end of the piazza, he only remembered things again on the right side, not the left. So in a sense, his memory was intact, but his ability to reconstruct was limited or disabled by the damage to his visual cortex.
Episode 200 of Brain Science is scheduled for September 2022, and I would love to include your feedback. If you want to be part of this milestone episode, just email me at brainsciencepodcast@gmail.com. to get more information. I will need all submissions by August 31st, 2022. I want to talk a little bit about some of the different parts of the brain that you discuss in your book, and I couldn't decide if I wanted to go from the bottom up or the top down, so I'm going to sort of hem and haw by starting at the top and then skipping back down to the bottom and then seeing where we end up in the middle. So I want to start with the neocortex and the question that I think is a real basic one, which is what is it that makes the neocortex different from the rest of the brain?
Okay, well, the neocortex is basically an invention of mammals. In animals prior to mammals, some of which are quite smart in a lot of ways, like birds, but they have very little neocortex. So this area of the brain that mammals invented in large mammals becomes by far the largest part of the brain. It completely surrounds and dominates all the rest of the brain that we refer to as subcortical. That's one aspect. The second aspect is that if you look in various subcortical parts of the brain or parts of the brain in non-mammalian vertebrates, every brain region looks quite different and has completely different types of cells and circuits. But the neocortex has pretty much the same six-layered sort of appearance everywhere throughout its extent. So it seems like once mammals stumbled upon this structure, they really liked it, and they elaborated it for lots of different purposes. So the neocortex in the motor cortex of a mouse would look a lot like the auditory neocortex of an elephant. It's 2 millimeters thick. It has the same 6 layers. It has the same sort of pyramidal cells and interneurons. So it seems to be a basic sort of toolkit for doing neural computation that is capable of doing lots of different neural computations. One advantage of using the structure for doing neural computation is that it may be possible to relate neural computation across very different parts of the brain in a much easier fashion than having very specific idiosyncratic circuits that are very different doing those kinds of neural computations. That's a second aspect of it. Its universality and its stereotype structure.
So that's why they could do that experiment where they took, I think it was a ferret, and rewired so that, I can't remember, was it the ears were connected to the auditory cortex or vice versa?
Yeah, I think that was done at MIT, and I think you can get visual input to go into auditory cortex and it'll function normally, or auditory input to go into transplanted visual cortex. So the neocortex works by what the inputs are. It processes those inputs and it sends outputs somewhere else, and you can transplant pieces of neocortex from pretty much anywhere to anywhere else, and it's the input-outputs that determine what it does, not anything that's highly dependent on the circuitry within the cortex, because the circuitry and what are called these mini columns seems to be very similar across the neocortex.
And that has important implications for if you're trying to recover from a stroke, that you might be able to use a different part of the cortex instead of the part that was damaged.
Exactly. And that seems to be important also for the evolution of animals as mammals expanded into various niches after the dinosaurs got blown away by the asteroid. If they wanted to be in a more auditory environment, they could just sort of grow more auditory cortex, or a more olfactory environment. So you had this nice toolkit that would work for everything, and it allowed them to expand and rapidly differentiate with enhanced skills. And as you say, it helps the process of recovery from the fact that the neocortex that's sort of near where you were damaged is a lot like the structure of the cortex that was damaged. And if you have a few parallel fibers going up, then you can get some recovery of function by just moving all the connections over a little bit.
And I think you said in the book that you could get more receptor density for a particular part of the body by having a larger area of cortex devoted to that, which we see in those famous drawings of the homunculus.
Right. Since the circuitry and the density of cells in the neocortex is about the same everywhere, what you find is that each square millimeter of neocortex is capable of processing about the same number of inputs. And so if you look at a map of the skin, for example, the sensory receptors are very dense on the fingertips and dense on the lips, forehead, several other places in the body. And so those areas of the neocortex are very large compared to the areas of neocortex that are devoted to, for example, the chest or the back of the legs, where the sensory receptor density is much lower. So that it ends up that each area of cortex has about the same number of sensory inputs.
And then the cortex, I guess, once we started having a lot of cortex, we could also have the development of lateralization, where one side of the brain would tend to do more of one job than maybe the other side. So I wanted to ask you to address the issue of the real differences between the right and left hemisphere compared to what we've heard.
Yeah, many books have been written about lateralization. The first thing, of course, is that at the most basic level, the right side of the brain generally controls and receives sensory input from the left side of the body and vice versa. So that part of lateralization is basically understood by everybody. But I think the thing that makes understanding lateralization so complicated is that in most people, particularly virtually all right-handed people and even the majority of left-handed people, the left side of the brain is really specialized for language in parts of it. And the right side seems to have some concomitant specialization for spatial processing. Now, it's argued by a number of people, for example, Gazzaniga, that these differences are differences in sort of style of processing that are important for other things, namely that the left side of the brain which is controlling your right hand, is important for sequential processing and things that require fine detail and analysis, whereas the right side is important for the holistic, broad picture, and also some aspects of novelty. Now you're getting into things that are sort of more metaphorical, that are a little bit hard to pin down, and it's not really clear which is the cart and which is the horse here. Was the left side always sort of stylistically more interested in sequential things and detail, and therefore language, when it got invented, had to go to the left side of the brain? Or did the left side of the brain become the way it is stylistically because it was forced to learn language? Language is hard to understand for a lot of reasons, but one of the ones is that no animal really has a true grammatical language like humans. Chimpanzees can learn lots of words and make up novel word pairs, but they don't, in general, pick up grammar very well. So you don't have an animal model for language. And it's also clear that language evolved very fast. Language may be only a couple 100,000 years old. That's short on the evolutionary time scale. When it arose, it must have been selected for very rapidly, because all humans easily acquire language and naturally in their childhood, unless there's something neurologically wrong with them. It's just an amazing organizational feature of the brain, its ability to acquire a language from just hearing examples of it. What the brain had to do to have this capability is the great mystery.
Just a comment about the whole left-right thing. Several years ago, I interviewed Brenda Milner, who you may remember spent many years working with HM. And I think she also did some work with some of Sperry's split brain patients. She pointed out to me when we were talking that in normal circumstances, the right and left hemisphere are in constant communication. So to try to say that this one does that and that one does something else is not necessarily a reflection of, normal function. I say that because, with Gazzaniga and Sperry, that a lot of that work was based on split brain patients who that meant they didn't have that communication anymore.
You're absolutely right. I mean, they do work together, and it's definitely a divide and conquer strategy. And doing a normal task, most normal tasks have both fine detail and large picture components, and your brain ships information back and forth, processing on left and right. I mean, that's exactly right. I agree.
Okay, so now I'm going to jump down to the other end of the story, because I particularly enjoyed your chapter about the structure and function of the spinal cord. It seems like it has a surprisingly complex structure, given that we pretty much share it with all other vertebrates.
Yeah, the spinal cord is a brain of its own right, and there is a lot of communication between the segments and the ability to generate locomotion patterns and control both the muscles within one limb. If you think about it, if you extend your limb, you have to contract some muscles and simultaneously relax other ones. And in reality, in general, you have to balance with your other limb and, in fact, your entire torso. The spine is actually doing a lot of this on its own much faster than it could do if it had to ship all the information up to the brain and have it shipped back down again. So it's a complicated computer in its own right.
There was one thing you talked about that I'm almost embarrassed to admit that this is something I've always been confused about. And even though it's a basic fact, I'm glad I finally have it straight. And that has to do with the unusual structure of the sensory neurons. I'm in particular thinking of what the axon does and the fact that the sensory neurons don't actually have dendrites.
Oh, the dorsal root ganglion cells. Yeah, so it's an interesting structure for sensory receptors that mediate touch and temperature, pain, and so forth in your skin. There's a neuron whose cell body sits just outside the spinal cord off the dorsal side in what's called the dorsal root ganglia. It has no dendrites, and it gives rise to a single axon that immediately splits. And one part of the axon goes out to the skin and forms little axonal endings, which are themselves transducers for the sensory input. So in other words, when the skin is pressed or whatever, that axonal ending fires action potentials. And those action potentials go in what we would think of as a retrograde direction toward the cell body, except that they bypass the cell body and go where the axon has split. And then they go orthograde in the other part of the axon and then synapse in the spinal cord gray area. They may synapse on relay cells that go off to the brain or participate in local reflex circuits and so forth. So they're kind of interesting cell, and that general pattern is duplicated all the way through the spinal cord.
Yeah, I've always been confused by the whole idea of, sensory axons. I've always thought of the dendrites as being the receiving guys and the axons as being the outgoing guys. And this is the one situation where the axon is kind of doing both jobs.
Right, and the nervous system is full of exceptions, or what a computer scientist would call a hack. This system seems to work well, and so it doesn't obey the classical picture of a neuron receiving stuff in its dendrites and shipping the information out on the axon, but there you have it.
Okay, we're getting a little bit tight on time. I always end up with more questions than I have time for, and I've only done the spinal cord and the neocortex and nothing in between yet. Of course, that leaves a reason for everybody to buy your book, but I'm going to give you a chance to pick something in between that you really pick something that you didn't know about that you learned because you had to write this book.
Okay. Particularly when I wrote the second one, I really wanted to learn more about the cerebellum and give a more coherent picture of what the cerebellum was doing in the whole motor control neural circuit for generating movement. And I came to appreciate, from my engineering background, the difference between feedback inhibition that's done in the spinal cord and feed-forward inhibition that's done in the cerebellum. In feedback inhibition, we could go back to the dorsal root ganglion cells, for example, some of which send receptors out that are reporting joint position, for example. So if you, say, hold your arm out straight for a while and then somebody puts a book in your hand, well, your arm will tend to droop. But if you're attempting to keep the arm at the same level, the droop angle is being measured, and you will have a reflex circuit that automatically generates more action potentials and the contractor muscles, and you'll compensate and keep the arm in the same position. So it's like a thermostat system for your furnace. If it's too hot, it turns the furnace off. If it's too cold, it turns the furnace back on. If your arm is too low, it turns the firing up and so forth. But for executing a complex movement, feedback inhibition isn't good enough and isn't fast enough. If you think about trying to hit a tennis ball, for example, you're swinging the racket and you have to make some adjustment based on, say, visual input that's happening very quickly. you don't really have a feedback system to use the information from. But what you do have is a cerebellum, and the cerebellum is a feed-forward controller in that what the cerebellum does when you practice a movement over and over again is build a model of what's happening in the movement and a model about what the response should be if the movement deviates from what the movement was supposed to be. Having a cerebellum, for example, allows you to thrust your fist out at a wall and stop exactly 1/4 inch in front of the wall every time with no error, even though you're having to decelerate and do a lot of complicated compensation based on your visual and tactile input before you actually ever touch the wall. Tactile input, I mean from your own joints and so forth. So the cerebellum is part of a very interesting system that does these very kinds of complicated computations involving building a model of the entire movement. One of the interesting things about that is that the cerebellum has a lot of cells in it. In fact, some people that work on the cerebellum claim that the cerebellum has as many cells in it as the entire rest of the brain, even though it's a structure most textbooks tend to not say very much about, because clinically, actually, if people have damage to the cerebellum, they become a little clumsy and are less able to execute fine movements, but they're often not so profoundly disabled. But you need a lot of cells to execute these really fine coordinated movements, and particularly to learn to execute fine coordinated movements. And what you find in evolution is that the cerebellum has increased in size alongside the neocortex. Rather than think of it as an old, archaic part of the brain that's just needed for primitive motion, it actually is an important part of the brain that's important for really fine skills. And it shows learning characteristics like other parts of the brain. Many types of motor learning are known to be mediated by changes in synaptic efficacy in the cerebellum.
But it doesn't have a cortical structure, though, like the neocortex.
It doesn't have a 6-layered cortical structure. No, it's not neocortex. It's way older than neocortex. It's as old as vertebrates. If, for example, you are a chess player and you imagine moving chess pieces, your cerebellum will be activated because those pieces have a movement component, and your understanding of how things move in 3D space is related to how you understand yourself how to move in 3D space, which ends up being coded in the cerebellum. So I really got to like the cerebellum after getting into it from my previous thought of, well, it was one of these old subcortical structures, and we'll do it with a spinal cord and then move on. So I like the cerebellum now.
And I suppose then the issue of does the limbic system exist or not would have been something you got a lot of feedback about from your first book?
You know, I tried to handle that issue very carefully because the term limbic system is still used and it still has usefulness, but the limbic system as originally described, going back to MacLean and the Papez circle, is not really a valid concept in terms of a system that is modular and works, internally according to its own function and then sends an output somewhere. If for no other reason than the hippocampus was supposed to be part of the original limbic system, but the hippocampus receives inputs from virtually the entire neocortex and has outputs to virtually the entire neocortex. So from that point of view, you can't go there. But the idea of a subcortical group of nuclei that do have a lot of modular interactions still has some validity. So I thought I handled that okay in the book, and I didn't really get any complaints about that. I did make some other mistakes, but not on that one, I think.
So is there anything else that you'd like to share before we finish up?
Well, I think the last thing is one of my current interests is in visual prosthetics, which I mentioned before. Most blindness is because of disease in the retina. Probably most of that is from photoreceptor death. You have several layers of cells in the retina, and even if the photoreceptor cells die, the retinal ganglion cells, which are the cells that I've studied most of my adult life, which are the ones who send their axons off to the brain, are still there, and they're sitting there on the surface of the retina. And if we could find some way to stimulate them electrically or optogenetically or magnetically or ultrasonically, there are a lot of people who have gone blind from macular degeneration or retinitis pigmentosa or other causes who could have some useful sight. And so I hope vision and neuroscience community can continue working and develop as much success with a retinal prosthesis as we now have with the cochlear prosthesis.
You mentioned at the beginning that one of the differences is that you need a lot more inputs to make a meaningful visual signal compared to the auditory. I mean, I talked to Michael Merznick about the original development of the cochlear implant, and he said, of course, back when they were first working on it, no one thought it would work because they didn't appreciate that the brain could begin to turn what at first sounded like meaningless noise into meaningful signals. I guess that was also because they didn't really appreciate how plastic the brain is. But the brain still's got to have a lot more information to work with to get some.
I think a typical cochlear implant has about 20 or 30 electrodes that are threaded into the cochlea. And the cochlea is tonotopic, which means that different positions respond to different frequencies. And you can stimulate those 20 different electrodes according to the frequency that's supposed to be there, which you know from the structure of the cochlea, and cause the auditory neurons to fire. That situation is very stable mechanically, and people are able to carry on conversations and talk on the phone and so forth.
Well, if we just think about how we can listen to an MP3 file, which is basically the auditory information has been compressed immensely, and we can't really compress video that way. That's one reason why video files are so much bigger, right?
Right, and they're two-dimensional, and the information is just denser to see any sort of pattern there. So you're talking about having to have hundreds of electrodes rather than 20, and you have to keep those 100 electrodes in close proximity to particular cells, like the retinal ganglion cells. And it's just been a much more difficult problem to take on.
But you've seen progress during your career, and you're hopeful there's going to be a lot more.
I think there's a lot of progress. I think there are optogenetic ways of stimulating these cells and electrical ways. There's tremendous progress in the development of computer chips that might be infrared light powered that could stimulate small numbers of cells. So I think a few of these schemes have been implanted in a few people, and I think within 10 or 15 years, things will be much better.
So if anybody that's listening to this podcast wanted to contact you, either to talk about the book or about learning more about the visual projects, how can they reach you?
Well, I'm in the Department of Psychology at the University of Alabama at Birmingham, and my email address is amthorfr@gmail.com.
Do you have your own website? I know the Dummies books have got some extras and all, but do you have a website of your own?
I don't have a website for neuroscience, actually. I do.
You have one for your art?
Yeah, I do a little bit of science fiction, futuristic art, and I have a Zazzle site and some other sites for that, but not for neuroscience.
Okay. So anything else you want my listeners to know about you?
No, I think that should do it. I'm happy.
Well, I enjoyed talking with you about this. I'm glad we had this opportunity to explore some of these ideas. You could go on for hours and hours, I know. But I want to tell you that I really did enjoy this book. It's funny, you think you don't want to read a book that's for dummies if you've been reading about something for years and years and years. But I actually learned some stuff I didn't know, as I revealed today.
Well, that's quite a compliment from someone.
So I think this book is one that, even though it's really intended for beginners, I think that it's got something for everybody.
Well, thank you. I enjoyed the interview very much.
I'll see you on the courts.
Okay, bye-bye.
I want to briefly review a few key ideas from this interview. But first, I want to emphasize that there's a lot of material in both books that we didn't have time to discuss. And this interview was about the Dummies book, not the Essentials textbook. There are discussions about how the senses, like vision, hearing, smell, taste, and touch, work, as well as the various parts of the brain in between the spinal cord and the cortex. If you like the Dummies book's approach, you will enjoy Frank's books. If you want more detail, then I recommend his textbook, Essentials of Modern Neuroscience, which came out in 2020. We started out by talking a little bit about neurons and what makes them special. We didn't actually talk much about the synapse, which is the gap between the neurons where a lot of important stuff happens. This discussion is in his book, but it's also in many other books that I've featured over the years. We talked about the difference between neurotransmitters and hormones. Dr. Amthor mentioned that some substances in the body can act as either one depending on where they are. A good example of this is epinephrine. The key difference is that neurotransmitters act in a more targeted way because they are released into the synapse and affect only nearby neurons. In contrast, hormones are released into the bloodstream and have more distant and diffuse targets. Of course, both require interaction with receptors, which means that what they do depends on which receptor they interact with. I want to emphasize this point because this is one reason why trying to create a drug that blocks or facilitates a particular neurotransmitter can have unintended side effects. That's why modern neuropharmacology is focusing on drugs that target the specific receptors instead. But the problem is we still don't know where all the receptors are located or what they do. The key take-home message is that it is really an oversimplification to identify any neurotransmitter with a particular action. Like calling dopamine the reward chemical is a gross oversimplification. We spent some time talking about the Nmda receptors, which are thought to be important in memory. These are fairly unique because they act like an AND gate. The Nmda receptor gate is usually blocked by a magnesium ion. This magnesium is only removed if an adjacent non-Nmda receptor is activated by glutamate, which then allows the Nmda receptor itself to be activated by another glutamate molecule. So essentially, two receptors have to be activated. This triggers a flux of both sodium and calcium. These receptors are thought to be important for mediating synaptic plasticity, so it's not surprising that the Nmda receptors are particularly abundant in the hippocampus, which is important to memory. Another thing we talked about was how brains are different from current computers. Dr. Amthor emphasized several points. The most obvious one is that our brains are massively parallel compared to computers, which usually operate serially. This means our brains are actually doing a lot of things at once, while computers do things in sequence, although this is usually not obvious because they are so much faster than our brains. Perhaps a more fundamental difference is that biological memory is reconstructive. There aren't any full memories stored in specific locations like they are in a computer. This is not a new idea for regular listeners, but many people still don't appreciate the dynamic nature of memory or its implications. The fact that memory involves recreating the context in which the original event occurred probably explains why when we go to certain locations, our memory for past events that happened at that place are triggered. I've talked in the past about how eyewitness memories can be altered so that they're no longer accurate. Things like leading questions can exaggerate this tendency. That's why we shouldn't assume that someone is lying if they remember an event differently than we do, or even if we think they've changed their story. As Dr. Amthor points out, you really can't distinguish which memory is right. Obviously, this reconstructive nature of memory has profound implications, especially in our judicial system. Right before I recorded this interview originally back in 2014, I had the opportunity to attend a talk by Elizabeth Loftus, who's probably the world's leading researcher in showing how volatile memory is and how easy it is to plant false memories. She did a demonstration during her talk where she showed us some pictures, and she was able to get at least half of us to misremember them. showing how easy this is to do. This was a really impressive demonstration, and I don't know how long it's going to be before we really deal with the implications of the reconstructive nature of memory. But returning to my conversation with Frank Amthor, we spent a few minutes focusing on the neocortex, which is the thin layer that covers the cerebral hemispheres. This is what distinguishes mammalian brains from other vertebrates, and many scientists believe that it is our massive neocortex that makes us so smart. Before the evolution of the neocortex, each brain structure was unique and pretty much committed to a specific dedicated function. With the emergence of the six-layered neocortex and its columnar structure, we have something that reminds me of the invention of the integrated circuit, because now we have a structure that can do different things depending on who it talks to. Obviously, whole books have been written about the importance of the structure of the neocortex, but Amthor does an excellent job of introducing this important concept. Consider the fact that much of the human neocortex consists of so-called association areas, where information from various parts of the brain are integrated. That's how we know that a particular sight and sound go together. This is possible because all these cortical areas are easily able to communicate with each other. Finally, we talked about the spinal cord, and Dr. Amthor said that it's a brain in its own right. He emphasized that even the spinal cord, which is virtually identical across vertebrates, is much more complex than we usually realize. He also talked about the importance of the cerebellum, which is another ancient structure that doesn't get much attention. Obviously, I've only hit on a few of the high points of our conversation, but I want to encourage you to share this episode with others and check out Neurobiology for Dummies and the second edition of Neuroscience for Dummies by Frank Amthor. The transcript for this episode is available for free at brainsciencepodcast.com, and you can send me feedback at brainsciencepodcast@gmail.com. Before I share a few brief announcements, I want to give a special shout out to my friend and guest, Dr. Frank Amthor. He is now retired from the University of Alabama at Birmingham, but I usually see him several times a week when I walk my dog Rusty around the tennis courts near my house. If you are looking for a good introductory textbook, I highly recommend the one he recently co-wrote, Essentials of Modern Neuroscience, from McGraw-Hill. This was episode 197. So if all goes according to schedule, episode 200 should be coming out in September 2022. To celebrate this important landmark, I'm inviting listeners to submit audio feedback for episode 200. This is something that I really enjoyed about episode 100. If you are interested, please email me at brainsciencepodcast@gmail.com. The deadline for submission is August 31st, 2022. I've already mentioned that listener support is essential because this podcast is independently produced. If you go to brainsciencepodcast.com/donations, you can compare the difference between the premium subscription and Patreon to find what fits your budget. However, I want to emphasize that even if you can't support Brain Science financially, you can help by sharing the show with others. You could even suggest me as a guest on your favorite podcast. Another way to support my work is to buy my book, Are You Sure? The Unconscious Origins of Certainty. It is available in paperback or ebook from Amazon and other online stores. If you want an autographed copy, email me at brainsciencepodcast@gmail.com. I hope you will visit brainsciencepodcast.com and remember to sign up for the free Brain Science newsletter so you can get show notes automatically every month. When you sign up, you get a free gift entitled 5 Things You Need to Know About Your Brain. You can also text Brain Science, all one word, to 55444. Brain Science, all one word, to 55444. I want to close out with a few words about this Encore episode. Encore episodes allow listeners to sample the back catalog of Brain Science, which goes back to December 2006. If you enjoyed this episode and would like full access to the back catalog, please consider becoming a premium subscriber. Thanks again for listening. I look forward to talking with you again next month. Brain Science is copyrighted by Virginia Campbell, MD. You may share the show with others, but for any other uses or derivatives, please write to brainsciencepodcast@gmail.com for permission. The theme music for Brain Science is Mind Fire, written and performed by Tony Catraccia. You can find his work at syncopationnow.com.